looking at my title now, I'm reminded of the phrase, the saying that all the really hard research questions have the really short titles. So we'll see how this one looks, shall we? So it'd be very important to note up front that this is work I'm doing with Mick Black, who's hiding at the back of the room, and also Phil Wilcox, who's hiding down in Dunedin. So, what we've been doing, well, the title's sort of summarising it, is working on, I said I'd be good and stand in front of the microphone, and I'm really not doing that. Maybe yeah, there we go. We've been developing some population simulations for testing different study designs. For anyone who did look at the abstract online, I'm going to slightly add a little extra of what was in there. I'm also going to talk about the computational side of things a bit, given the nature of this meeting. Okay, so a little bit of the genomic background for a start. When people are saying we found a gene linked to cancer or something, what they're doing is what's called GWAS, genome-wide association studies, where you get genomic data from many, many people, and then you're looking to see which of these genetic sites, these SNPs, as I'll be referring them to as, are associated with your trait of interest. And then from that, you can find all the SNPs that are passing some statistical significance threshold and use that to predict risk for a trait. No, okay, I have to be a little careful with that. And these risks are increasingly being used, like in the real world, to actually give people predictions of their risk for diseases. The downside is that all these big data sets that GWAS, that PRS is done on, are primarily Europeans. And if you're not a European, you might have unique differences in your genetics that are just not being accounted for. Now, ideally, you'd just repeat the same kind of thing in New Zealand, get a nice big data set and find some more appropriate PRS GWAS estimates. But, well, it's expensive. I probably don't need to talk about sciences funding in New Zealand to anyone. And also, many Māori have, for a variety of historical reasons, pretty negative feelings about taking part in such a study. So instead, okay, I still don't know where I'm meant to be pointing that, that's right. We have to try something else. I feel like did I skip a slide there? Yes, so something else which doesn't involve on 10,000, 100,000 genotype people will need to be done instead. Okay, I think the clicker's not my friend today. So, if we're not just relying on really big genomic information, what could we do instead? Well, Māori have really well recorded whakapapa, genealogy information, going back many, many generations. And what we could do, is there somewhere in particular I should be pointing the clicker, or is it just, it thinks for a moment? Ka pai. We can use this known genealogy, this whakapapa, to estimate how related individuals are, which is giving us sort of a genetic correlation between them, how much of their genomic identity is shared. And happily, methods exist that you can use this. You can use these known co-ancestries. You can use this with available trait information, and so instead of getting GWAS of big data, PRS to predict your risks, you can combine whakapapa information with what you have to predict it in a different way. And this exact thing has been used in selective breeding in livestock, in crop improvement for many, many years. So, more modern approaches, I'm going somewhere with this, this is the preamble. More modern approaches are adapting this by using some genomic information. So 15-ish years ago, 17 years ago, GLUB was proposed, which is estimating the co-ancestry between individuals based on genomic information. There's also a more modern approach called SSGLUB, which is estimating it from your full whakapapa combined with some genomic information. Ooh, I've been upgraded, okay. There's a big button that says laser. I'll have to push that to find out what it does. Oh, fantastic. And so what we're doing in our project, sort of the focus, the framework behind it is what value does traditional Māori knowledge have for improving and giving new options for disease prediction, for risk prediction. Rather than just relying on genomic things, which as I've mentioned isn't really a one-size-fits-all solution, how can we make use of whakapapa and what is it offering us? So, towards that, we need to think about what good study design would be. How many individuals, if wanting to use this SSG blot method, how many individuals would be needed, what genomic data would be needed, and who is available to be genotyping. And so towards that, what I've been doing with Mick's help, with Phil's help, is developing a whole bunch of simulations. Using some very slick software, which is pre-published, which I'll talk about it in a bit more, starting back from Western Polynesia and evolving, migrating to East Polynesia and to Aotearoa, to end up with, hopefully, a somewhat realistic modern-day set of genomic Māori information that we can use for testing different study designs on. Oh, double-clicked it, okay. Yeah, there we go. So, a quick historical oversight. So the people who, the Lapita people, moved to Western Polynesia thousands of years ago, were there for a long time, migrated to Eastern Polynesia, and then about 800-ish years ago, the first people migrated to Aotearoa. There's then a period 100 years before Europeans, but a couple of hundred years ago, Europeans evolved, and that's not quite right, arrived. There's here are some historical events, which you can read about in history books and are unpleasant, but are incorporated into our simulation details. And then we wind up with the modern day with, hopefully, eventually about 1 million Māori, although we'll have to scale up to that. One particular detail I'll highlight on that we'll come back to later is that when we're simulating Aotearoa, you might have to use your imagination a little bit, we're representing it as a big 2D space. So individuals are born, married, die, etc. in different locations in this space. And the slick bit is that we can do things with this. What happens is that we're stochastically simulating and arise naturally different clusters of individuals, say 1, 2, 3, 4, and these are representing abstract, artificial, iwi and hapū. So just by how we set up the simulation and tuning the details, we can simulate these abstract groups representing extant population structure. I'll have to come back to that later as well, though. So, the microphone, okay. Right, so now for the actual more computing side of it, which is that research is hard. What we're doing here is bringing together several different areas. I've got a big quantitative genetics background, which is the SSG blood model prediction side. We're also using population evolution for our simulation, and then translating it all into actually predicting disease in people is much more a precision medicine thing, linking genes to traits. So because of this, with a range of field areas has come a range of hassles. So, firstly, the data itself, the genomic data that we are simulating, it gets really big. So the software SLIM that we're using is really well optimized for calculating how inheritance happens, going from one generation to the next. It does a really slick thing with haplotypes and haplotype libraries, for those who that means things to. But our output files are still really large. That's not a particularly technical way of putting it. So instead of using VCF, which is a more standard format for storing genotype data, because it's simulated and it's just going straight from the simulation to the model fitting, I'm using a very succinct format that's much smaller. So about 10,000 individuals of 1.5 million SNPs, 1.5 million different genomic sites of variation, is about just under 20 gigabytes. Now we're wanting to scale this up to 1 million individuals, as I mentioned before, so there'd be about 2 terabytes, which is like not impossibly unmanageable, but because it's simulation, because it's stochastic, it's not based on real data, you want to do a whole bunch of repeated runs so you can get a range of variables and more confidence in your results. And so the numbers start getting big, or at least big enough that NeSI is going to have some strongly worded emails for me. Now, unfortunately, flow on from this is because we're using this non-standard data format, lots of the standard processing things, Plink, VCF tools, various R packages, they don't match it. On this MS data format, that's all just basically zeros and ones and nothing else. You can't use them. Further, because as I mentioned, this is sort of multi-field, and I'm trying to move from the population evolution format to quantitative genetic software, there's also not pre-existing software that will translate between those for me. Happily, I suppose, although it took long enough, a variety of me actually having to sit down and do some C coding myself got there. But I thought I'd mention this both as a challenge that existed and also because I don't get to grumble enough, I suppose. So the third sort of challenge I'll touch upon here is that also, because we're multi-field, the software I'm using is really good, but we're not using it in really a typical use case. It's really well optimized for standard population evolution kind of questions, which are along the lines of, with much generalization, you have running many generations with a finite, smallish population, saying how do the seabirds on this island evolve if we give them 200,000 years? What we are doing is sort of flipping that around. We have many fewer generations, only going back, say, 1,000, 1,300 years or so, but we want to have a really big population, getting up to 1 million people. And this leads to some unusual, or maybe not expected by the developers, problems when I'm running this simulation. So I mentioned before our spatial element to Aotearoa. We end up with these nice clusters of individuals. I had a smart idea when I did that, which was that to have two mirrored individuals deciding who's marrying who, that's going to be inversely dependent on distance. If someone's really far away, you're less likely to have children with them. The downside of this is that it means every single generation, when it's calculating these probabilities, it's quadratic. That is an order that's quadratic to the number of individuals. And so if you have 500 individuals, that's fine. But if you have 500,000 or 1 million individuals, then it's having to calculate, I shouldn't have tried to do maths on the spot, getting up towards near 1 billion different probabilities every, is that right? I'm sure that that's near enough, and you'll forgive me if it's not, 1 billion different calculations just to work out your marriage probabilities each generation. So, what to do? Well, cutting the Gordian knot, maybe not the most elegant solution to this, was just to limit the range that individuals can get married at. So I suppose we've got, ooh, that guy right there, I'm wobbling a bit. We restrict it so that only individuals within a certain range can be considered with marriage, which drastically cuts down the number of possible marriage partners and makes it much faster. Okay, good. Sort of fixed that one. The last challenge that I'll be talking about is that actually fitting the models themselves. So, got through all the simulation, have this simulated data, it's been processed, it's all ready to go. Now what? So I mentioned up the top, and I'll expand on them here in a little bit more later, 3 different models we're sort of considering. There's BLUP, which is using full whakapapa information for prediction, based on these co-ancestries, these kinships. There's GBLUP, which uses full genomic information, so supposing we genotyped every single person. And then there's SSGBLUP, which uses full whakapapa information, but where it's available, it'll augment that with available genomic information. And we're wanting to fit these with BLUP and GBLUP, which you can think of as 0% genotyped and 100% genotyped, and then SSGBLUP with 10, 50, and 90%. So we're getting a range of how many people need to be genotyped, basically, and we can look at performance as we vary that. Now, the tricky bit is anything that's using this genomic data for just the way the stats models work out, you need to invert a really big genomic relationship matrix, which is quantifying these kinship values I mentioned, which looks something along the lines of this. You've got a diagonal, which is my hand stable enough, which is the individual's relationship to itself, so these are all near 1, and then the off-diagonals, these guys aren't really related, these guys 0.5, so maybe that's like a parent-child relationship where they're sharing about half their genetic material, and so on, all based on the genomics. This matrix sucks, honestly. It's big, it's non-sparse, it doesn't have any zeros in it, and so trying to invert it is really difficult. People have been iterating and improving and trying to do better ways of finding the inverse of this, well, ever since SSGBLUP and GBLUP were proposed, basically, but it's still a bit complicated, it's still not easy. There have been definitely improvements. People have written some really slick software, much better than anything I'd write. But ultimately, to get above a certain point, you just need better hardware. Oh boy, okay. So, I feel like this sounds like the point where I'd have an answer for this. I don't, unfortunately. You just have to deal with it as you can. Right, so putting aside from the computational things, getting back to actually sort of the results in a big picture sense, which took a little while. We're focusing on what value, what Māori knowledge, what traditional knowledge is giving us when we're predicting disease. If we're wanting to move away from purely full genomic things which aren't great, aren't suitable in all cases, and want to make use of traditionally known information, what does that give us? Well, I'm going to actually flip this around a little bit and say, conversely, what value does genomic data give us? If this whakapapa information is there and is accessible, what point is there in actually genotyping things? What is that giving us? What advantage is there? And so we can look. Jumped ahead of myself. So as I mentioned, we're genotyping different proportions. So known genotypes all the way up to 100 with some intermediate values, and the model changes slightly over that range. And we can look at how the X3 changes with more individuals genotyped. And we see, and I hope the text should be available to make out, that, unsurprisingly, as we get more genotype information, we have, I'll stop waving that around, it's going to be distracting. We're seeing the true actual genomic values rather than things based on relationships, based on pedigrees, based on expected things rather than directly measuring DNA. And so with more DNA, we're seeing better values. But we're seeing that they're not that much better. We're getting 60% accuracy just purely from whakapapa and 80%, dear me, I am going to blind some, 80% when everyone's been genotyped. But with, say, 50% of the population genotyped, we're most of the way there. And 90% genotyped is almost the same as genotyping everyone. And genotyping is expensive, and we can see diminishing returns. So to sum up, because I'm about out of time, it's hard doing new things. No, that's easy to say, isn't it? Trying to move away from a sort of standard paradigm of these GWAS, find SNPs associated with a trait, use that to predict polygenic risk scores, has brought a whole bunch of challenges, sort of computational. But we do see that there's value in it, right? These are showing that the whakapapa information is really useful for prediction. And probably more importantly, it's allowing us different options. We're not stuck in a sort of one-size-fits-all way for prediction. So it's worth it. Thank you.
Thanks, Alastair. I think we have time for one or two questions, perhaps. Let's start with gentleman here.
Oh yeah, thanks for the talk. I have to admit this may be a naive question because I'm not a bioinformatician, but it relates to the large matrix inversion, the non-sparse one. How exact does the inverse need to be? If you could deal with reduced precision for a faster computation, would that work?
It depends is never a good answer to give, but it depends a little bit. So there's different ways that it can be solved. There's ways that solve it directly, but what people tend to do is use either slight approximations, there's this thing called matrix bending, amongst other things, or they use iterative approaches, which converge towards your thing. And then that's just sort of a matter of setting your threshold for how precise you want to be. It matters some, but not that much.